So now we're going to take a little bit of a turn, and we're going to look at some of the science that's driving all of this innovation. Of course, the science needs homes, which we've been talking about. But our guests here are going to talk a little bit about one of the key areas of science driving innovation, which is genomics, genome editing, and the new CRISPR technologies that have made such advancements in medicine possible. So Alex, why don't you introduce yourself, and then we can talk about that.
Sure, thanks Nancy, and thanks for joining all of us here at NYC Builds Bio Plus 2022 Life Sciences Innovation and Development Symposium. I'm Alex Philippidis, Senior Business Editor at Genetic Engineering and Biotechnology News, joined by Premsrirut, an MD PhD, who is founder and CEO of Mirimus. It's an interesting Brooklyn-based company that works to push the boundaries of CRISPR, RNA interference, and PCR testing technologies. And you'll hear more about all those. Our focus these next few minutes is on approaches to gene therapy developed over the past decade known as genome editing. Those new technologies have seen growing venture capital investment at a time when the dollars and numbers of deals in gene therapy and even CRISPR have declined year over year. Just a small example, and I won't flood you with numbers, in genome editing a year ago, third quarter, no deals, no companies funded. In the third quarter of 2022, actually 3 companies in the third quarter, and almost $8 million in venture financing. Doesn't sound like a lot, but at least they're going in an up direction at a time when other fields in the broader biotech are going down in investment. Before I go further, I ought to define, I think, the terms I'll be using. According to the National Library of Medicine, gene therapy is a medical approach that treats or prevents disease by correcting an underlying genetic problem. For example, a faulty gene with a mutation. At first, gene therapy was limited to transferring or adding a gene into a patient by introducing a new gene into cells to fight a disease, or a healthy copy of a gene to replace a gene that causes disease. Now, that gene is usually enclosed within a carrier or vector, such as an AAV. You may have heard that term. That's an adeno-associated virus. Over the past decade, however, newer approaches have emerged. Several of them are forms of genome editing in which molecular tools are introduced into cells to change the existing DNA. Among those forms of genome editing, things you've heard about, in CRISPR, researchers treat disease by removing, adding to, or altering sections of the DNA sequence. CRISPR uses a pre-made piece of RNA or guide located within a longer RNA piece or scaffold. That scaffold binds to the DNA while the pre-designed RNA sequence guides an enzyme to the right part of the genome. And that enzyme could be Cas9 or something that's smaller or more recently discovered like a Cas12 or 14. Over the past 6 years, base editing has come into use by researchers. Now, base editing uses CRISPR-Cas9 to modify genomes by introducing point mutations in DNA without double-stranded breaks. And in July, Verve Therapeutics became the first company to dose its first patient with a first base editing therapy that reached the clinic. It's called Verve 101, and it's designed to treat heterozygous familial hypercholesterolemia, or HeFH for short. Another gene editing approach, or prime editing, can be thought of as a genetic form of search and replace. It can perform targeted small insertions, deletions, and base swapping precisely in a target DNA sequence or a donor DNA template without making double-stranded breaks. And earlier this month, a prime editing therapy developer called Prime Medicine raised $175 million in gross proceeds through an initial public offering, or IPO. Now, since then, the price of their stock, well, it started at the IPO price of 17. Yesterday, it was trading around 19, which is no small feat in these days of stock declines for most biotechs. Several companies have emerged in recent years to capitalize on genome editing technologies, and the ones that are furthest along in clinical trials are those that specialize in CRISPR therapies. Researchers and investors will be watching closely over the next few months at these companies. Closest at CRISPR Therapeutics, with which its partner Vertex Pharmaceuticals are expected in November to begin FDA submissions seeking rolling review and approval for their CRISPR therapy called exa-cel. It's designed to treat two blood disorders, sickle-cell disease and transfusion-dependent beta thalassemia. The submission is expected to be completed in the first quarter of 2023. And the two companies have released positive data over the last couple of years after earlier trials. The first patient receiving a therapy, a woman named Victoria Gray, who's gone public, has stayed free of sickle cell disease for nearly 2 years. Intellia Therapeutics and partner Regeneron Pharmaceuticals last month released positive data from a phase 1 study of NTLA-2001. It's a one-dose treatment for ATTR, that's transthyretin amyloidosis, or a buildup of abnormal proteins on the heart that can cause heart failure. Editas Medicine earlier this spring introduced a new CEO, and by year's end is expected to come out with a clinical update for their lead candidate, EDIT-101, an in vivo CRISPR-edited medicine. In vivo means it's administered in the body directly. They don't take cells out. For this EDIT-101 is for a rare eye disease called LCA10, Leber congenital amaurosis type 10. And in April, Editas began the first dosing of a pediatric patient, a kid, with this EDIT-101. So that's a short lay of the land for genome editing. It's my pleasure to welcome Premsrirut, the founder and CEO of Mirimus. And as you'll hear more about, her company develops animal models using CRISPR and RNA interference, or RNAi. And as she'll explain, Mirimus also develops RNA therapeutics and carries out high-volume COVID-19 PCR testing. Let's welcome Prem.
Thank you so much. It's a pleasure to be here, and that's a really hard lineup to go up against. And talking about our small company, which has been around for about 12 years, and even though we've been around for 12 years, I still feel like, well, we still are considered a startup, especially in this environment here where we're doing a lot of research and development and still working towards that initial therapeutic that we'd like to take to the clinic. Just to give you a little history of Mirimus, we actually spun out of Cold Spring Harbor Laboratory based on the RNA interference technologies. So it predated CRISPR, and that was a little scary part, is that we launched about 2 years before CRISPR even was discovered, or I would say first published on. And with RNA interference, it kind of took the trajectory pre-CRISPR the same. It was all a lot of hype, a lot of investment into it, and it was meant to be the next therapeutic phenomenon, because we could actually target any gene of interest and silence it without actually touching the genome itself, because it would silence the RNA and prevent protein from being expressed. So we took this technology into animal models, and we were building new systems and could show that we could turn genes off and on in animals. And we were developing that right to go into therapeutics. We launched this company. And 2 years later, CRISPR came out, and all of the investments in RNAi just sort of fled away. And so we were sort of stuck there, wondering what to do next. But with CRISPR, you can learn it. You can see the amazing developments that you can do with it. And as mentioned, you can really target a specific gene, both DNA, RNA, and perform a lot of the editing that can fix thousands of different familial diseases. What our company does is we specialize in the preclinical animal model development. And so if we work with companies such as Roche, Novartis, and so forth to develop their preclinical platforms and utilize the CRISPR platform as a tool, we can now actually generate cancer models in both mice, rats. And what used to take about 4 or 5 years to even set up that development, we can do this in 4 to 6 weeks. And by using CRISPR and actually electroporating into the animal in vivo, in a specific tissue, we can generate precise genomic edits that will mimic the human disease, and it'll give rise to that cancer model. So this dramatically speeds up the way we can develop drugs. We can test new theories, hypotheses, mechanism of action. We can also build these animals to have human genes. So that way we can actually test the drug within the animal, because in many cases, the animals, when you're going through with rodents and even non-rodent animals, the human gene that the drug is hitting is not the same as in the animal. So we'll have to do what's called humanization of that animal. So we use a lot of these models, and we do speed up the research that we do to take a drug to the clinic. We are still now working on our RNAi therapeutic system, and as mentioned with gene therapy, this would be a gene therapy coupled with AAV delivery. The downside of RNAi is how do you get it to the place you want to and not to the places you don't want it to go. So it's that off target, the toxicities that we're worried about, and we've still been developing this. And over the last decade, there's been so much improvement on AAV delivery systems, gene therapy, and we believe coupling our technology with a lot of these new systems will get us there. So we're currently partnered with Biogen in developing new RNAi therapeutics to treat neuromuscular diseases. And lastly, as we've really expanded, through COVID, we have had one of the most challenges in terms of, being in the heart of Brooklyn, hit very hard, and just helping our community. And we realized that the lack of testing, the inability to build the assays quickly at a time when all of these reagents were just unavailable. We were there to help the hospital, SUNY Downstate, as they shifted into one of 3 COVID-only hospitals in the state. We kind of did a call to action and called all of our team in to help build the assays that were necessary. So through all of this, over the last 2 years, we've performed over 5 million COVID tests. We've kept about a third, we've done testing for about a third of Westchester County public schools, and about 25% of all New Jersey public schools, as well as a lot of the independent schools within the New York and the tri-state area. So we're really proud to have grown that team, and we've realized what it means to have a lack of available space at a time of need when your company is expanding very rapidly.
So you're located at SUNY Downstate right now, and you have more than one location, right? So how many employees do you have?
So we have about 100 employees. Mm-hmm. We were, when the pandemic started, we were probably in about 2,500 square feet of space in an incubator right next to SUNY Downstate. And suddenly we took another 1,000 square feet, and then we took a floor of another building adjacent that's also managed by SUNY Downstate, the incubator. And then we had to take an office building down the road. And during the height of the pandemic, I have to admit, I had to take an apartment right behind the lab because I couldn't make my way home about 5 or 6 days a week. So that became my office. It's directly behind the lab. And we also have another about 1,500 square feet or so within SUNY Downstate because we do utilize their vivarium. So currently we are about 28,000 square feet spread across 4 or 5 different buildings. Mm-hmm. And it is extremely challenging to coordinate. So we are looking for that really nice space that's being built to consolidate all of our operations.
But you're in a special zone, right, for your employees, so that makes it a little bit more difficult.
It is. With all of this new space being built, one of the great things about being where we are is we are a part of the New York Startup Program. So all of our employees do have access to the tax-free income, state and city tax, as well as our company has also privilege to that on corporate taxes, which we realized is a once you actually do start generating revenue, it is a big factor.
And does that help you recruit?
It does help us recruit, but we do find the challenges of recruiting in New York City in general. We get a lot of the young population that's there that wants to come in, but the turnover is quite high. Mm-hmm. Recruiting the population, fresh postdocs with young children is always a challenge in the city where they're always thinking about the schools and access to good public school education.
So just to put all of this in a little bit of context, Alex, what started this whole kind of genome revolution was the completion of the Human Genome Project, which was the first sequence of a human genome, which basically would help you to understand what your genetic makeup was. And the next step was kind of to apply that to what they call precision medicine, which allows you to get targeted therapies based on your genetic makeup. And then in 2013, a pair of women scientists, Jennifer Doudna and
Charpentier, yeah.
Published the first CRISPR studies, and they were subsequently awarded the Nobel Prize. And as you can tell from what Prem said, this set off a sea change in the field in terms of the approaches, and companies had to pivot and new investments had to be made. But there is enormous amount of progress being made in the field, as Alex just shared with us, with lots of different companies all over the country and the world, really, using these tools.
The challenge is getting these therapies both in a form where they're going to be proven safe and effective, then getting approved, but even more challenging is getting them to patients. There's issues of access. You know, there's still a big question about how are these going to be priced. Gene therapies, as we've seen, have been priced as much as $3 million for Zynteglo. I believe it's, no, the second one, or close to it, of Bluebird Bio. And it is thought that, well, maybe the CRISPR-edited therapies can be offered for less, but we'll see. We don't know that because companies love to recoup years of R&D expenses by setting a price for the drug that can be pretty high in many cases. Now, the counter argument you'll always hear is, well, we have access programs, we have ways through usually private setups, but again, patients have to go through a process and show to somebody they really need it. And these are still pretty pricey medicines and not really subsidized. So in that sense, how's that going to be worked out? That's still to be determined. Also, you see the kinds of therapies out there. Yeah, by next year, some form of CRISPR therapy is going to be approved, it is expected. But you have other treatments up the pike. Prime Medicines, for example, has a pipeline of 18 different therapies, Prime edited. Only about half of them are in what they call priority areas, I&E disorders, liver disorders, and blood disorders. But that's still going to take them years to first get into the clinic, let alone get through it with trials. So.
Well, thank you for helping us understand a little bit more about this area of the science that's driving the growth and development. Thank you.
Cool. Thank you.